Hi, welcome to this video from Swansea University Chemistry Department and Zimmer & Peacock. This presentation was first given as a workshop at CIGES in Spain 2022 as part of a biosensor conference, and now we've moved the contents onto the YouTube video as part of our promotion of Swansea University. So what I want to do is I want to sort of essentially jump straight into it and talk about the three elements that are covered in this workshop. We'll talk about introduction to voltammetric biosensors, amperometric biosensors, potentiometric biosensors, and impedance biosensors. So we'll talk about things like the detection of capsaicin in chili, glucose. We'll talk about pH, and we'll talk about things like immunosensors and molecular diagnostics. We'll give some real-world examples. And if you were part of the original workshop that was held in Spain in 2022, then you would have actually been part of the live demos as well. That said, I'm just going to do a quick introduction. This is the Zimmer & Peacock team. As of today, this team is actually 85 people in total. If you're just wondering who I am, you can find me either at Swansea University or at Zimmer & Peacock, and you can also find me at LinkedIn. Just to say, Zimmer & Peacock is a contract developer, contract manufacturer. We have world-class facilities in Norway, both an R&D and a manufacturing facility. And we also have locations in the UK, in Coventry, and also on campus at Swansea University. If some of what I say today, you want some more material on this, you can also visit the CP Academy, and there's a couple of free courses on the Academy that you are very welcome to look at. And we also have other videos on the Zimmer & Peacock YouTube channel, which may be more specific. There's one on, for example, wearable biosensors, and there's one on impedance spectroscopy. We also have something that's called the Developers Zone, where we try to answer questions from biosensor developers. So now I'm going to sort of jump into the into really this main part of this presentation. So at Zimmer & Peacock and also at Swansea University, I on occasions lecture at Swansea University, and I lecture about electrochemical biosensors. So electrochemical biosensors really fall under the sort of overall umbrella of electrochemistry. So electrochemistry is often used in the study of corrosion. These days it historically has been the science, let's say, of batteries and energy and power, but because of obviously there's a move to electrification, portable power supplies, etc., then really one of the biggest applications these days of electrochemistry is in, for example, lithium-ion, lithium polymer battery development. Also, electrochemistry is used in a lot of electrochemical synthesis processes. So, for example, most of the aluminium in the world is produced by what's called the bauxite process, and that's a large-scale electrochemical process. Now, at Zimmer & Peacock, and also as I say through some of the lectures that I do at Swansea University, there's an analytical chemistry, and underneath analytical chemistry you could have this sort of branch of electroanalysis. And then, as I say, what we do at Zimmer & Peacock is sensors and biosensors, and that sort of falls under the category of electroanalysis. So I'm now going to sort of jump into electroanalytical chemistry and sort of say that there are many techniques in electroanalytical chemistry. There's voltammetry, so there's cyclic voltammetry, which I will talk about a lot today, but that's also to say that but it shouldn't ignore the fact that there's also square wave voltammetry and differential pulse voltammetry. I will talk about amperometry today. I would say that amperometry is a technique that's widely used in the glucose detection space, but if I was developing glucose sensors today, I wouldn't necessarily use amperometry. Amperometry is a technique that was simple enough to implement some decades ago, but these days with modern electronics, you really don't need to have such a simple waveform, but we will discuss it. Potentiometry we will discuss today. Potentiometry is also a technique that's extremely widely used in our world. The most measured chemical parameter is probably pH. Most pH meters in a laboratory will actually be potentiometric in nature. So it is something that a lot of scientists and engineers are familiar with, or at least use, for example, whenever they make a pH measurement. And we'll also talk about impedance spectroscopy. Now, this is not something that's been used so much in commercial applications of, let's say, medical diagnostics, but I think it will increasingly be so. And again, it's the modern electronics that make this actually possible. So going into this a little bit, I think one of the first things to do is if you're an undergraduate or graduate and this is the kind of equipment that you're used to, where you have a kind of, the potentiostat itself. This one's from CH Instruments. You have the electrochemical cell, which is here a glass with the electrodes in it, and obviously, potentially a laptop to drive all of this. This is the way electrochemistry was done up until 5 to 10 years ago. But nowadays, electrochemistry is actually much more of a modern science. You're able to have these thumb-sized potentiostats, which can actually be used with things like smartphones, at ZP, and at other manufacturers as well. You will also find these sort of handheld or hand-sized potentiostats as well. And also something that's also modernizing, let's say, at least electroanalytical chemistry, is the availability of screen-printed electrodes. Now, I have to make a disclaimer here that at Zimmer & Peacock, we do manufacture screen-printed electrodes, and they are extremely high quality. I think there's a lot of screen-printed electrode manufacturers out there. You have to be a little bit careful because the variability from electrode to electrode can leave people with a bad impression on screen-printed electrodes. But when they're made well at high quality, then they really are excellent and repeatable and as good as, and I'll touch upon this, things like glassy carbon electrodes. So I will go through this. I don't want to say quicker, but I will go through this in essentially more detail now. The first technique I'm going to jump into is voltammetry, and as I say, I'm going to sort of focus on cyclic voltammetry today. So this is a an animation or a simulation of a cyclic voltammogram. This one is of ferrocyanide. If I just get it to run again, I'll sort of break down what we're actually looking at. So what we're having on this y-axis is the current. So we're looking at a current, and we're looking at the way it changes with voltage. So in this experiment, what's happening is that the voltage is being changed, and the current from the electrochemical system is responding accordingly. Now what we're actually looking at is the oxidation of ferrocyanide to ferricyanide, and we're looking at the reduction back again. I will break this down a lot more. We're going to keep coming back to this in several ways, so I think the repetition will help with the understanding of it. But this is one of the kind of classics, that if people were to have an iconic image of electrochemistry, at least analytical electrochemistry, it's probably cyclic voltammetry, and often you'll have these kind of voltammograms used. Now what we're looking at here is the waveform that is generating this voltammogram. I just said that we change voltage, we look at current. So the voltage is changing with time. So we start at 1 voltage, we go up to another voltage, and then we return to it. It's almost like a triangular waveform, and that is a term. A triangular waveform is used a lot in science, and it is also used in this branch of science. So what's happening here is we have a start voltage. In this case, we have a start voltage of minus 500 millivolts. We go up to another voltage. In this case, the voltage is 500 millivolts, and then we return to the original voltage. Now that does seem complicated, but I want to say that these experiments are not that hard. You take something like a screen printed electrode from, dare I say it, Zimmer & Peacock. You take one of these potentiostats, you put the ferrocyanide solution, it might be something like 10 millimolar ferrocyanide in 1 molar potassium chloride. You put that on the electrode, you run the software, and you will get one of these voltammograms. And to do this experiment takes sort of 2 minutes, if not less. So it's hard to say, hard to explain, but actually doing electrochemistry or electroanalytical chemistry can actually be quite straightforward, as long as you've got the equipment, the necessary equipment. I'll refer to this later on, but we call this an E mechanism. And the reason it's called an E mechanism, it's quite simple. It's just a electrochemical step. I'll show you something called an Ece mechanism in a bit, where there's an electrochemical step, there's a chemical step, and then there's another electrochemical step. So the E, you'll see this sometimes in literature, there'll be EE, which is an electrochemical step followed by another electrochemical step, or you'll see EC prime, which is electrochemistry followed by a catalytic chemical process. This is just E, and I will touch upon an ECE. So there's all these kind of nomenclatures, and they sort of tell you the sequence of electrochemistry, chemistry, electrochemistry, for example. So diving into this a bit more, why does this voltammogram look the way it does? So there's, it's an unusual shape, and I will say that when people are teaching electrochemistry, they get really hung up on this shape. I would say tell you in a bit that this shape is really just a function of the way the experiment is done. Though people think it's terribly important, I think they overemphasize the importance. But we will explain what we're seeing here. So we have sort of an area of the voltammogram here, which is really a region where you're applying voltage, for example, to the ferrocyanide, but there's not enough voltage to actually cause an oxidation of ferrocyanide. So we just have a background, almost capacitive current. Then we hit a region where actually the voltage is reaching a sort of a threshold voltage, where actually we're starting to get the transfer of electrons of ferrocyanide to the electrode, and the ferro is converted to ferri, and we start getting current, and we call this our Faradaic current. This term Faradaic or Faraday will turn up a lot in electrochemistry, and it's in homage to Michael Faraday. You can see how old electrochemistry is from this image here, but Michael Faraday was one of the kind of founders, I would almost say, of modern, at least chemistry, and definitely one of the founders of electrochemistry. So we have this, but going back to the cyclovoltammogram, we have this region which is flat, which is really a reflection that we're not getting, they call it sometimes non-Faradaic, we're not getting any electron transfer. We hit a critical voltage and we start getting electron transfer, and the current starts increasing. Now there's more regions in this Faradaic region. There's sub-regions, and there's the first sub-region here is what we call almost kinetic control. Oops-a-daisy. So what's happening in kinetic control is we're just starting to apply voltage, and the voltage is causing a current to flow, and that current is really dictated by something called the Tafel equation, and we're under kinetic control. What it means is the current is exponentially proportional to the voltage. So theoretically, you could just keep increasing the voltage, and the current would just keep on going up. But there's other, I want to say, physics that kicks in that stops that being reality. We don't just get infinite current with infinite voltage, because there's other processes that kick in. But at the foot of this, let's say, this wave or this curve, we have the current is proportional to the voltage applied. So current is proportional to voltage. Now there's another region here which we call the mixed control. And in the mixed control, you're not able to just keep on getting more current just because you're applying more voltage. You're starting to actually deplete the ferrocyanide at the electrode. The ferrocyanide is having to diffuse in from the bulk, and it simply can't keep up, or the diffusion process is not fast enough to keep up with the electron transfer, and you start getting a depletion of ferrocyanide near the electrode. So I've shown here concentration as a distance from the electrode. So for example, in our scenario, we have ferrocyanide at a concentration of something like 5 millimolar in the bulk, and as we go from the kinetic control region and we start going into this mixed control region, we keep driving the electron transfer of ferrocyanide that we're actually starting to deplete the ferrocyanide. So it's simply not 5 millimolar near the surface anymore. It's starting to decrease, and the more voltage we apply, the bigger that depletion zone becomes, and so we start building up this depletion zone, and we start getting what's called a diffusion gradient. So at some point, and the point is at the peak, the current is no longer linked to the voltage, because in fact the reaction is going as fast as it possibly can, and it's limited now by diffusion, and so we hit a sort of new region here, where the current is actually proportional to, you would hope it was proportional to concentration. I should really maybe talk about that in a minute, but it's also proportional to the scan rate. So I will just stop and sort of do a quick summary. So when you're taught, or you hear people talking about cyclovoltammetry, they think it's terribly, terribly important. Cyclovoltammetry is interesting, and it is analytically useful because things like the peak current are proportional to a concentration. So there is an analytical value in this. I can say my peak height is this, therefore my concentration is this. So that is useful. You must also understand that actually the shape of this is also the shape and the currents we're looking at are also a function of the way the experiment is done. So we will talk about this in a minute, but the faster I scan the voltage, the higher this peak height. You will see in a lot of electrochemical papers, actually lots of graphs where they show peak current is proportional to the square root of scan rate, and it's always in there. But the real value add to society is not that high. All it's telling you is that, yeah, they have a signal, and it's proportional, and it's controlled by diffusion. That's interesting, but it shouldn't be overemphasized how important that is. That's just, it's not a fundamental signature of quality of experiments necessarily. It's just quite an easy experiment to do, and so people essentially do it. But we go from our background region, where the voltage is not enough to cause electron transfer. We start coming to our kinetic region, where actually current is proportional to the voltage, but you drive that reaction so fast that in fact diffusion starts becoming the limiting step, and you get to this kind of peak current where it's all under diffusion control. Now I just made a comment that this peak height in cyclovoltammetry is proportional to the scan rate. So here I'm showing you that as we scan, there's also a sort of, as we scan along, we get a voltammogram as a result of it. And I did explain that, in this region, we're essentially in our background region because the voltage is not near any kind of critical value. Then we start coming up to our, I'll just back up slightly. And then we come to our sort of kinetic control region, which is kind of essentially here when you look at the voltammogram. Then we come up to our mixed control region, and then we come up to our diffusion control region. And we start getting this iconic peak and then this fall off. And the reason we get the fall off is because we're literally just overdriving the reaction now with the voltage, and the diffusion can't keep up, and we're just starting to increase this depletion zone. And that's why I said it earlier on, and you'll see this in a lot of electrochemistry papers, where the peak height in a cyclovoltammogram is often proportional to the rate at which you drive the voltage sweep. So the faster the voltage sweep, the higher the peak current. So here's the sort of the voltage, and as that increases, or the voltage sweep increases, the peak current also increases. So you could ask yourself, well, what's the significance of this, and why is this coming about? So what's happening is this: we have this ferrocyanide that, as I said, is in the bulk, or is in the solution at 5 millimolar. At this peak, what's happening is we are depleting the ferrocyanide. So if we scan very fast, what that means is this: that you you get from voltage 1 to voltage 2 very quickly. And so you hardly spend any time at any of these voltages, which means that you don't consume all the ferrocyanide. But as you decrease the scan rate, the whole experiment takes longer. And that longer means that actually you're spending more time converting ferrocyanide to ferricyanide, and we get these concentration gradients. And these concentration gradients, this is the electrode itself, and we have distance from electrode. So when I scan fast, I do my experiment very quickly. I hardly consume any ferrocyanide, and I have a very sharp concentration gradient. But if I scan the voltage very slowly, what's happening is I'm depleting ferrocyanide, and I get a much, I deplete it, and my concentration gradient actually gets shallower and shallower and shallower the slower and slower I get. Because what's happening is this, that this diffusion layer is quite large when I scan slowly and quite narrow when I scan quite fast. And so this distance here is part of the equations. When I've like this, when I've scanned slowly, I have a sort of depletion or diffusion layer that's like this. When I scan very fast, I have a diffusion or depletion layer that's more like this. So what's happening is this distance is controlling this gradient. So what's happening is that my current is proportional to my difference in concentration, which is 5 millimolar because that's the bulk concentration, 0 millimolar because I've depleted all the ferrocyanide at the surface, and it's actually divided by this diffusion layer distance. But I said that if you scan slowly, the number's quite big, and if you scan fast, the number is small. And so when you scan fast, d is small, so 5 divided by a small number is quite a big number. When you scan slowly, d is bigger, so 5 divided by a bigger number, the gradient is therefore lower. And so all that's happening is as we change the scan rate or increase the scan rate, this gradient is getting steeper and steeper and steeper. And as that gradient becomes steeper and steeper and steeper, the current, the peak current, becomes higher because that current is really a reflection of this diffusion gradient. So I know you might have to sort of rewind this several times, but electrochemists think this is really important, really iconic, that their peak current is proportional to scan rate. It's interesting and it is published a lot, and they kind of consider it that it's really fundamental. As a pragmatic biosensor developer, I think it's interesting, but it's not absolutely critical. It could be a metric that you're doing your experiments in a fairly good way. But the reason for it is that when you scan fast, your diffusion layer is quite small, and therefore your gradient is quite high. When you scan slowly, your diffusion layer is, the distance is, let's say, bigger, and therefore your gradient is smaller, and therefore your current appears to be smaller as well. And this is why in the Randall-Sevic's equation, current is proportional to the square root of scan rate. I wanted to say, and I've said it several times, that electrochemists find this very fundamental, but the reason I don't find it quite so fundamental is if you did the same experiment but in a different manner. So when you look at an iconic cyclic voltammogram like this, it's done in an unstirred solution. But there's just as well, you could do it on what's called a rotating disc electrode. If you're a biosensor developer, I don't recommend rotating disc electrodes necessarily, certainly not outside of an academic setting. And the reason being is they're nice for characterizing mechanisms and understanding mechanisms, but if you're trying to make it like a handheld point of care device or a continuous glucose monitor, it's a distraction of characterizing, or it distracts you and you're characterizing this system, but you're not really adding a lot of value. But the reason I bring up rotating disc electrodes and hydrodynamic voltammetry is this. The cyclovoltammetry is iconic in electrochemistry. If you did a very similar experiment, again, you could do it on 5 millimolar ferrocyanide, but you were doing it on an electrode that was rotating. What you're now doing is you'll keep on replenishing the material at the electrode. So rather than getting these diffusion and these diffusion layers and these depletion zones, you don't, because you keep on replenishing the material. So if I look at this literature results, if I do a rotating disc electrode at 100 RPM, so it sounds quite fast, but it's turning away. I get something that's a bit like normal voltammetry, that I have a background, I have a region that looks like my mixed, I have my peak current, and then I have this kind of drop down again. Now there's a second process that's happening here, and that's why it goes back up again. But that looks a bit like the voltammetry that we've just been explaining. Now, as you increase the rotation rate, so for example, I go to 1500 RPM, it changes its shape. And the reason it changes its shape is both it gets higher and it changes its shape. Now, the reason it changes its shape is because we don't get this depletion zone, because every time we consume the ferrocyanide, we're spinning fast and we're just bringing in more and more ferrocyanide. So we don't get this depletion. And the reason the current is higher is because now the current's under sort of essentially what we call mass transport control. The faster I rotate, or therefore the faster I pump, the faster I bring solution, the higher the current. And so now we're under mass transport in this case. Now, the reason I bring this up is because now you understand that cyclovoltammetry is just a reflection of an unstirred reaction. And if I stir it, I get a different one. So why is, let's say, rotating disc electrode work not just as iconic as the voltammetry? And really it's just because voltammetry came first. So the reason I bring it up is because I do see people who are doing biosensor development getting very hung up whether their voltammetry necessarily fits all the ideal parameters, and whether it does or doesn't is not so important. What's important in the biosensor development world is whether your assay is proportional to concentration, and what's your sensitivity, what's your selectivity. It's not whether your voltammetry is actually iconic or not. So, of course, this iconic shape I've been saying in voltammetry, especially this depletion, is all due to this depletion zone or diffusion zone here, and it goes away in rotating disc electrodes because we're actually stirring essentially the experiment and replenishing the ferrocyanide. And in this case, we have I think it's the Levitch equation. Current is proportional to the rotation rate, or the square root of the rotation rate. And so, my whole point on this last slide was just to say, cyclovoltammetry, though it's iconic, is just a function of an unstirred reaction. And if you're a practical, pragmatic biosensor developer or electrochemical sensor developer, don't get so hung up on it, I would say. Now, we started this conversation on voltammetry by talking about ferrocyanide, and I said that was an E mechanism. This is a molecule called capsaicin. It's the molecule that's in chili products, and what we're showing here is that capsaicin has a very different voltammogram to just the simple ferrocyanide that I was showing earlier on. So what I'll do is I'll let this voltammogram essentially restart. I will explain that this is showing you what's called an Ece mechanism. And so as it starts now, what's happening here is we have our background signal where we're not at any threshold voltage, and now we do get a oxidation, and then we get a reduction. So having an oxidation reduction, so you sort of oxidize something and now you're reducing something. Now what you'll notice is on the second scan we get something different. There's an extra peak. So that's quite interesting. So now we'll sort of dive into this. The reason I put this slide up here is to show you that voltammetry is interesting. This is almost like a fingerprint. It's very distinctive. There's actually, in total, there are 3 peaks, but the third one only appears on scan 2. So if you were trying to make an assay, you can see that you can really take advantage of this, that, you know, these peak heights are actually proportional to the molecule of interest, and the appearance of the third peak on the second scan is very indicative that this is really capsaicin. Let's jump into this a little bit more. So what's happening in the first peak? What we have is the oxidation of the capsaicin to a, now I apologize, I'm a chemist, but it's what's called a cation radical. Now what's not obvious necessarily to you is that that cation radical is not stable and there's an immediate reaction with water. So electrochemistry is a kind of unusual analytical science that actually you're analyzing this molecule, but in the process of analyzing it, you're actually allowing it to go into another a chemical reaction. So we have the detection of the molecule in this first peak. We form a cation radical, and then actually it's also undergoing a reaction with water, and we're forming a new molecule that's called a quinone. So this is the quinone molecule. Now, what's happening in our reduction wave or reduction peak is we're reducing this quinone to quinol. Now, what's happening on the when we get the third peak on the second scan is, I'll just do this very quickly, this quinol is actually being oxidized back to the quinone, because the quinone, or the quinol rather, was not in the reaction at the beginning. So we actually ended up forming, we took capsaicin, we detected it, we through hydrolysis formed a quinone. The quinone got reduced to a quinol, and then we see the quinol on the second scan, but it's a new molecule, and that's why it only appears once we've done the first cyclovoltammogram. So that's quite a distinctive fingerprint for that molecule, and we can call that an Ece mechanism. So as I say, the E stands for the first electrochemical oxidation, the C stands for the chemical reaction, which was hydrolysis, and then we have a new molecule, so we have another electrochemical processes as well. So we call that one an Ece mechanism. Now, if you were with us when we first did this lecture and webinar in CGES, and we actually did a demonstration here on voltammetry. So these were the attendees. We had about 40 attendees, and we actually demonstrated this live. I mention this because if you have an opportunity to see Zimmer and Peacock at a conference, you know, we don't just give dull workshops. We actually do live demos as well. So hopefully you'll be able to catch us at a conference or one of our workshops. So I'm going to dive now into ion-selective electrodes. We just spent some time talking about voltammetry. Now I'm going to talk about ion-selective electrodes. And before you say, no, I've not heard of ion-selective electrodes, there's just about no scientist, be it a biology, a material scientist, a chemist, you know, practical physicists, you know, everyone at some point may have used a pH meter. The world has gotten a lot more modern, and so we are able to measure ion-selective electrodes. Ion-selective electrodes, I just sort of jumped into that term a bit, but you have potentiometry, which is the measurement of voltage. And in sensing, we often use that with a subgroup of sensors called ion-selective electrodes. You can have an ion-selective electrode for nitrate, potassium, sodium, chloride. I mentioned these because these are all the ones that we have on our DP web store. But pH is the most widely used ion-selective electrode. So when you pick up a pH probe in a lab, it's often got a pH-sensitive glass bulb on it. So if you look at it very carefully, you'll somewhere find on it some glass, and that glass is actually pH sensitive, and so you're measuring that, the voltage on that glass surface versus a reference electrode, which is often silver chloride. And I'm just saying that at the moment, you know, pH meters and pH electrodes are quite big and bulky. I think there's going to be a real change because actually this stuff doesn't have to be that big. At Zimmer and Peacock, we make a solid state pH electrode, which is, you know, very, very small. it's almost the size of the, well, not almost, is the size of the electrode shown in this image. I did a lot of explaining of the, let's say, the shape or the theory behind voltammograms. I'm not going to do that so much on potentiometry, but I do want you to know that if I use the example of a pH sensor, there's the signal, which is really a voltage, and it's proportional to the log of the analyte of interest. So in the case of pH sensors, voltage is proportional to the log of protons. In the case of a nitrate sensor, voltage is proportional to the log of nitrate anions. In the case of sodium, voltage is proportional to the log of sodium cations. Now, how these things work is we have a reference electrode, and this reference electrode is intended to have a voltage that's independent of the sample. So it's kind of isolated from the sample, it's not influenced by the sample. And we measure that voltage versus an ion-selective electrode. Now, an ion-selective electrode often has a property or a molecule associated with called an ionophore. So the classic one for potassium is valinomycin. Valinomycin is a circular molecule that the potassium cation can fit in with and snugly fit in there. And so we call the valinomycin the ionophore, and the potassium is the ion that can enter or leave essentially that hole, which is the valinomycin. So we have the second electrode that has an ionophore of interest, and we measure that ionophore, or that electrode versus the reference electrode. And so this is like the ZP. Many of our ion selective electrodes actually look like this, where we have a working electrode that has the ionophore of interest, and we measure the voltage here versus the voltage here, which is our silver chloride reference electrode, and we just measure the voltage like that. Now, as I said, in CJES, we were actually, when we first gave this webinar, again, we don't just make people sit there, we actually get them up, and here you can see an image of one of our engineers called Siobhan doing a demonstration for the attendees on ion selective electrodes. So I said at the beginning of this this video, this lecture, this webinar, that we were going to do voltammetry, which we have discussed. Now we've done potentiometry. Now I'm going to jump into amperometry, which is, in a commercial sense, is probably along with potentiometry. So potentiometry is commercially used a lot in pH sensors. Amperometry is used a lot in glucose sensors. So in amperometry, I said in voltammetry, you had that sort of triangular waveform, or at least in cyclovoltammetry, you have a triangular waveform. Amperometry is almost a simpler cousin of it, that we don't have a triangular waveform. What we do is we have, and I'll show it here, we just have voltage, and we just hold voltage stable throughout the experiment. So for example, if I was using an oxidase enzyme, enzymes, glucose oxidase, lactate oxidase, alcohol oxidase, if I was using one of these oxidase enzymes, they produce hydrogen peroxide. You then want to detect the hydrogen peroxide, and you use a voltage of something like 650 millivolts. It's a bit like earlier on, actually. When you're doing amperometry, if you stir and you have mass transport, you will have one current and a particular shape in your signal. And if you don't stir, you could have what we call the Cottrell response, where actually it's a bit unusual for science, because I think in science we often expect sort of steady state signals, just a signal that's just kind of flat. But in amperometry, actually, you get a signal that falls with time. This fall with time, we kind of call it the Cottrell response. It's governed by the Cottrell equation. It's the same reason in voltammetry, where you have the iconic voltammogram, you come up to a peak and then it starts falling down because of a depletion zone, which is diffusion controlled. And the same thing can happen in amperometry. If you just have a, let's call it a glucose strip, and you turn it on at 650 millivolts, the signal will jump up and then start falling with time as you start to consume all the analyte. So you have a sort of initial, and then the current, as I say, falls with time, and it's governed by, it's characterized by the Cottrell equation. I would say governed by the Cottrell equation. And the current is proportional to the concentration, so it's a useful analytical technique. As I say, it's most widely used in amperometry. It's most widely used in the glucose space. I mean, at Zimmer and Peacock, we have what we call a generation 2 and a generation 1 glucose sensor. I'm just going to do a quick analytical sketch of a glucose sensor. So we have often maybe something like a carbon electrode. We'll have a mediator on top of that. A mediator is a molecule that's able to shuttle or carry electrons between, let's say, a chemical reaction and the electrode. So it's mediating. Ferrocyanide are mediators. Ruthenium hexamine is a mediator. Ferrocene is a mediator. Methylene blue is a mediator. Modula blue is a mediator. There's lots of mediators out there. These are basically chemicals that are able to take up or give away an electron, and they're able to shuttle electrons between chemical reactions and electrodes. So we have a mediator, we might have an enzyme layer. So the enzyme is what gives the specificity, and then we might have a barrier layer, and I'll explain the barrier layer in a minute. So it's like this. So I'm going to describe with a practical example glucose. So I have glucose. Now glucose in real samples is actually quite high. I know the word 5 millimolar sounds like a low number, millimolar. But in biological terms, it's actually quite high concentration. So we have this glucose that's at fairly high concentration, and it's not very hard for that kind of concentration to overwhelm the enzyme. We simply can't get enough enzyme down to keep up with all that glucose. So we have glucose. The glucose diffuses to the barrier layer, but the job of the barrier layer, one of its jobs is to actually block some of the glucose so that not all the glucose gets to the enzyme layer. And in the enzyme layer, we then have a enzyme, for example, glucose oxidase, which is actually reacting with the glucose. And the enzyme itself happens to get reduced, so it happens to retrieve electrons, or receive electrons rather, from the glucose. The glucose actually forms gluconic acid. The enzyme itself gets reduced. No, not gluconic acid, glucolactone. It's the glucolactone then hydrolyzes to gluconic acid. But in some ways, I don't want to get hung up on what happens to the glucose. I actually want to talk about now that the glucose is reacting with the enzyme's picking up the electrodes. The enzyme is essentially trapped with these electrons. But what happens is it has a natural mediator. So for example, oxygen, which reacts with the enzyme, forms hydrogen peroxide, and then the job of the mediator, for example, is to recycle the hydrogen peroxide back to oxygen, and the mediator picks up the electrons, gives up electrons, so the mediator goes from reduced to oxidized, and then we recycle the mediator at the electrode, and we get electrons flowing. So there's a lot of material, not a lot, but there's some, here I'm showing at least 3 zones in the glucose sensor, or 4 zones. We have the electrode, we have the mediator, we have the enzyme layer, and we have the barrier layer. The barrier layer one of its jobs is to stop all of the glucose necessarily getting to the enzyme layer. The enzyme layer is to undergo a specific reaction then with the glucose, which ultimately forms hydrogen peroxide. And the mediator layer is to allow us to shuttle electrons from the hydrogen peroxide directly to the electrode. The reason people do use mediators is because you can then essentially detect the hydrogen peroxide at lower voltages, maybe 250 millivolts. And we would call that a generation 2 glucose sensor, because we are using a mediator to help us in the detection of the glucose in that case. In a generation 1 sensor, there is no mediator, and in fact we're detecting hydrogen peroxide directly. The only issue with the generation 1 glucose sensors is then that you're using 650 millivolts, which means that you can accidentally detect ascorbic acid, or maybe uric acid, or maybe acetaminophen. So you do have more interferences at these higher voltages. So a generation 1 glucose sensor is simpler to make. That's a real advantage in commercial world. But then you suffer the downside of increased interferences because you're actually using a higher voltage. Whether you go for generation 1 or generation 2 glucose sensor really depends on the application that you're actually trying to face. And that's really almost like a commercial decision. And that's one of the important things I think at least at Zimmer & Peacock is there's the commercial balance versus the technical performance. If you're having technical performance, you have to sometimes pay for it, and you've got to make sure that the end user can actually afford this. Again, we would have when we give this, we don't just sit there and tell you about this. We actually do a live demonstration, and so if you do get an opportunity to come to one of our workshops, then we'll do a live demonstration of this. Now, finally, I'm going to touch upon electrochemical impedance spectroscopy, but I start off with a question, which is why. The reason I start off with a question, which is why, is this: that impedance spectroscopy is taught too often from the perspective that it's intrinsically important, you should know about it. But I want to tell you why you should know about it before I actually, So what I'm showing you here is a output of some impedance some impedance spectroscopy. What we've done is this: we've taken a Zimmer Peacock carbon electrode and we've put a series of antibodies on the surface of that electrode, and then what we've been doing is we've been testing the electrode with what's called the antigen, and as we add antigen, the signal changes. So as we go from 5 nanograms per milliliter, we get one signal. 25 nanograms per milliliter, the signal changes. 50, the signal changes. 75, the signal changes. What you'll notice is this: that we've actually done this experiment in triplicate. So there's essentially something like 12 data points here at 4 concentrations, and each concentration is tested by 3 electrodes. I bring this up because these were handmade sensors, and often in academic settings, people are trying to make very reproducible sensors. To make really very reproducible sensors by hand is a real skill, and at ZP we can do it because we really have world-class equipment. We do it a lot. Our environment is very well controlled. Our humidities, our temperatures, our dust, everything's very well controlled, so we can do it. So I want to say to people, don't be too hard on yourselves if you're suffering. We're trying to do reproducible sensors in an academic setting. It's not easy when that environment and your equipment is not so much under your control as it would be in a commercial organization. But the reason I'm saying the why is this: the why you should be interested in impedance spectroscopy. It's quite a good technique for doing what we call immunoassays. So if you're interested in detecting proteins, IL-6 is a protein that's is an inflammatory marker that's implemented in, or is a marker for sepsis. So for example, people are also PSA, prostate specific antigen, troponin I. I'm just listing out proteins that have medical and therefore and commercial interest. When you're trying to detect proteins, immunosensors are actually quite good, and a good way of using an immunosensor is actually impedance spectroscopy. So I'm trying to answer the question why. There's also now an increase in molecular assays. There's a lot of assays in the literature for SARS-CoV-2 where they have they're looking for the genetic material from SARS. They've immobilized a capture, we call a sort of capture strand on the surface, and when these two things bind, we get a signal. So impedance spectroscopy is very good at molecular assays and immunoassays, and the reason you should care about them is that these are the kind of assays that help in human health, for example. So impedance spectroscopy is often taught for the sake of impedance spectroscopy. I want to tell you that it's actually a good technique for doing medical diagnostics, especially for proteins and especially for genetic material. So we have our 5 nanogram signal, our 25 nanogram per milliliter signal, our 50 nanogram per milliliter signal, and finally our 75 nanogram per milliliter signal. So you can get some nice data from this kind of stuff. Now I'm going to jump into the fundamentals of spectroscopy. Now that I've attempted to justify why you should be interested, I'll now sort of talk about it a little bit. So impedance spectroscopy is a good technique for looking at interfaces. And so when you make things like immunosensors, you have an interface where you've put for example, an antibody on it. and when the antigen binds to it, the interface is changing. And so impedance spectroscopy is a really good technique for actually measuring that amount of change, and you hope that amount of change is proportional to the analyte of interest. So the way impedance spectroscopy works is it's using Ohm's law, and Ohm's law is this simple, that the current that you see flowing at an electrode is proportional to the voltage that you apply divided by the resistance. And I'm hoping that this resistance changes as you bind and unbind the molecules of interest to your surface. Now I can investigate the resistance because, for example, I could measure the current at 1 voltage, change the voltage, measure the current again, and from there I could tell you the resistance of the surface. And in physics, this kind of stuff is often taught in physics, where they just have a simple resistor. You can put a voltage across the resistor, measure the current, and it'll tell you the resistance of the resistor. And in this case, for example, it's 5 ohms. Now, I'm going to specifically talk about Faradaic impedance spectroscopy. I'm not going to be talking about non-Faradaic impedance spectroscopy. I prefer Faradaic impedance spectroscopy. You might have to look up the definition, but in Faradaic impedance spectroscopy, I've put a molecule in there that I know is electrochemically active, and I want to watch how that molecule's interaction with the surface changes as I get binding. So I prefer Faradaic impedance spectroscopy. So in Faradaic impedance spectroscopy, I have a voltage, and on that voltage, I put a little amplitude. I just changed the voltage just slightly. And what this means is that I get as I slightly change the voltage, I slightly change the current. And as that current slightly changes, you know from Ohm's law that I'm applying voltage, I've got a current, I can therefore calculate resistance. So at any one time, I'm applying a voltage, but I'm just putting a little perturbation on top of that voltage. By the way, when I say the equipment does this really well for you, so there's no really any hassle. And the current is also changing, and then the software just actually can calculate you the impedance by just seeing the amplitude of the current. So you don't have to be, the software is actually doing a lot of this for you. Now, there's something interesting in electrochemistry that actually a surface, when you have an electrode in a solution, it's acting partly as a resistor and partly as a capacitor. If the surface is acting purely as a resistor, as you change the voltage, the current is exactly matching it. That's a pure resistor. There's no lag. I change voltage, current's changing. I change voltage, current's changing. Just completely sort of one for one. They're overlaid. If a surface has a capacitive element to it, and a capacitor element means the surface is trying to act slightly resistive. And so the surfaces in electrochemistry, especially immunosensors, for example, actually have a capacitive element to them. And it's because of this. You have an electrode, you're putting proteins on that surface. Those are like biological polymers. You're slightly insulating the surface. So the surfaces don't act like resistors. They act like a bit like a resistor and a bit like a capacitor. Now what that means is, as I change the voltage, the current is changing. But capacitors have the slight ability to store charge. And so everything is offset slightly. I change the voltage, the current changes a little bit later. I change the voltage, the current changes a little bit later. And the bigger the capacitive element, the bigger the delay. Again, the software does this essentially for you, and it will tell you what these capacitive elements are. So I suppose one of the things about impedance spectroscopy is it's a powerful technique. The software is powerful, and people have put decades of research into it, so you can just benefit from it, essentially. So we have our electrode, and when it's in solution, so for example, I put a solution here, the surface is acting actually like a lot of tiny little capacitive and resistive circuits. They actually call this, I have a resistor here, I have a resistor here, and I have a capacitor here. They call this sort of Randall circuit, and it's a model that is not a bad starting point for describing the surface. Now I'm going to go back to a chemistry model. So for a second here. So this is the situation. I have my electrode, and I'm trying to oxidize ferrocyanide to ferricyanide. This is my counter electrode here. And I can model this system using called a Randall's, they call it Randall cell here. I've also see it called a Randall circuit. And these capacitors and resistance symbols kind of mean something. This resistance of solution is the resistance between the working and auxiliary electrode, and that shows up here. We also have the what's called the double layer capacitance. This is the alignment of ions on the surface, and that appears as this capacitor here. And then we have the ease at which electrons can transfer from ferro to ferri, and we call that the charge transfer resistance. Now, when you're doing an immunoassay, you're binding to the surface, and so your signal could either be appearing in the capacitive element here, or it could be appearing in the charge transfer resistance element here, or actually it could be appearing in both. Now, what impedance spectroscopy is doing is, I said that to you that impedance spectroscopy is able to calculate the impedance of the surface because we're changing the voltage and measuring the current. Impedance spectroscopy actually allows you to play with the frequency. And so what happens is this, that if I apply a fairly low frequency, my current passes through this element of the circuit. I go through the solution because I can't ignore it, and I go through my charge transfer. The reason being is at low frequency, capacitors actually have high impedance or high resistance. As I go up in to like 1 megahertz, for example, whoopsie daisy, as I go up to 1 megahertz, for example, the current prefers to go through the capacitor. The capacitor has a low resistance / impedance at high frequency, and then when I'm at mixed frequency, intermediate frequency, like 1 kilohertz, you end up going through both. So the power of software in a modern spectrometer is this, that you're able to run an experiment, and you might run the experiment, you might say, right, my DC voltage is 650 millivolts. On top of that, I might put an amplitude of 5 millivolts. So I've got a fixed voltage of 650 millivolts. On top of that, I put a little amplitude on top of that. I may set that to 5 millivolts. And then I'm going to investigate my surface using a frequency of something like 1 hertz or 1 cycle per second to 100,000 hertz or 100 kilohertz. And what the software, what you're able to then do is actually take those signals, come up with what's called the equivalent circuit, and actually examine these elements as a function of concentration. So I show some data where I went 5 nanograms per milliliter up to about 75 nanograms per milliliter. Because these elements, especially the capacitive element and the charge transfer resistance, will change the more you bind to the surface. So yeah, a capacitor has a, I sort of said it, and this is sort of the electronic circuit that says a capacitor has a impedance that's inversely proportional to the frequency. So it's easy to push current through a capacitor if you have high frequency, and it's much more resistive or harder if you're at low frequency. And so we can use frequency to help us understand both the capacitive nature of our surface and our resistance nature of the surface. And the only reason we care is because we're hoping that we've made a surface that's specific to the protein, and as the protein binds or unbinds, we can use EIS, impedance spectroscopy, electrochemical impedance spectroscopy, to understand the coverage at any particular concentration of the surface by that particular protein. So here's an example. We take a gold electrode, we functionalize it with a sort of single strand of DNA. Somebody then has the matching DNA, which might come from like E. coli, for example. And as we change the amount of DNA, it's called the Nyquist plot. I haven't explained the Nyquist plot, but it's often these semicircles. These semicircles are changing their circumference as we get more and more binding. And then to understand that, we can actually form an equivalent circuit for that. And those elements in the equivalent circuit, things like the charge transfer resistance, are proportional to the amount of binding. And essentially, it's a way of saying, take a gold electrode, make it specific to the analyte of interest, test it with the analyte of interest, and then you'll find that things like the charge transfer resistance are proportional to the binding, and therefore you've made yourself a molecular assay. And this is what we sort of essentially did here. And this is just a zoom in on the previous slide. Slight comment here that historically people have done immunoassays and molecular assays. They often use gold electrodes. They use gold electrodes because they like to form self-assemble monolayers. These are where thiol atoms are binding to the gold, and then you attach to the sulfur atom is also a sort of carbon chain, and they all stack up quite neatly. And the gold sulfur bond is quite strong. So there's a lot of comments there. And what I want to say quickly is that gold is a rare, fairly rare commodity. There's a lot of industries that want gold, and therefore actually it's a bit of a premium material. These days I'd actually recommend people use carbon. There's a molecule called pyrene NHS. We do have it on the ZP website. It's quite a good molecule for coupling antibodies to carbon surfaces, by sort of telling people these days to start looking at carbon as a default go-to material rather than gold, because gold at the time of making this video it's really spiking in price. And for example, ZP can end up spending something like $50,000 on gold, and I just don't think universities can necessarily afford this kind of material. To make the gold electrodes is not so hard. You take a gold surface, you put a thiol molecule on it. A sort of go-to molecule in this kind of application is mercaptohexanoic acid. Whoops a daisy. You then activate something like the carboxylic acid with some simple coupling chemistry, and then you can attach antibodies to that. And then the last thing you want to do is block that surface to stop non-specific binding. BSA is often used for bovine albumin, and we do have a gold electrode at ZP that we sort of use for this kind of work. Whoops a daisy. What I want to do is then show you here, we used the gold 303 in this particular work, for example. Now carbon is my more preferred material to go to these days. It's probably, there's not much limitation on the amount of carbon that we can get in this world. There's quite a nice paper that we were authors on. It's probably worth a look because it's quite a simple fabrication procedure. As I say, there's a molecule in this paper called pyrene NHS. It seems to be in short supply at the moment, so you might have to get it off the ZP web store, but hopefully it'll become more available. Its lack of availability at the moment is actually linked to many things. That at the time of doing this video, there's unfortunately supply chains. Whoops a daisy. Supply chains are sort of very messed up in the world, and so even chemicals now are actually becoming a little bit hard to get hold of. But what we do is we take the carbon surface, we functionalize it with a molecule called pyrene NHS. It's really quite simple to do. You just drop cast it down, and then you can actually just put the antibody on there, and it'll sort of essentially stick the antibody on. And then we block the molecule, block the surface with chitosan. And we often follow these we follow our functionalization steps using things like cyclical voltammetry. So if you're following the paper, we sort of see that we can change the surface, and the cyclical voltammetry becomes more distorted the more functional materials we add to the surface, because we're essentially blocking the surface. We can then test that surface. We have our surface that's functionalized with the antibody. We have a reporter molecule in here. I think in the paper it's something like ferricyanide. And we then test. We do what's called differential pulse voltammetry. I talked about cyclical voltammetry, or differential pulse voltammetry is a more modern, I would say, methodology. And when you're doing analytical electrochemistry, I think the side of voltammetry is fine, but actually the methods I would actually recommend are square wave voltammetry and differential pulse voltammetry and impedance spectroscopy. My preferred technique is actually impedance spectroscopy, but in this paper we use differential pulse voltammetry, and what happens is as we change the surface, the signal changes accordingly, and this is literally the results out of the paper, and we've been able to repeat this, quite a few times on different assays, so it's quite a go-to, let's say, method for us. It's probably just worth saying that, if we put something like 111 hours into an assay, we can often make things work. I'm not trying to tell you this is the best data in the world. I can see a few problems with our limit of detection, but it's just to kind of say that making immunoassays is not a 8-hour effort. We would optimize, and we do this quite routinely. We could spend something like 111 hours easily on this, and I know at university sometimes as part of a PhD, you're very much on a learning curve, and people take a lot longer than this. I'm going to slightly change gears, but I suppose before I change gears, I'm going to do a quick summary. So this talk so far, which is in part with Swansea University, we've talked about electrochemistry, and under that electrochemistry, we've talked about electroanalytical chemistry. Underneath electroanalytical chemistry, we talked about voltammetry, potentiometry, amperometry, and impedance spectroscopy. So I've said that voltammetry, for example, it can be used for the detection of capsaicin. Amperometry is often used with enzyme sensors, for example, glucose sensors. In potentiometry, for example, is used in ion-selective electrodes, and that's pH. And then I've said that impedance spectroscopy, electrochemical impedance spectroscopy, is often used in what we call immunosensors and molecular assay sensors, somewhere where you've got a macromolecule on a surface and something is binding to it and changing the surface. I'm going to change gears slightly now and just sort of talk finally about practical troubleshooting tricks and tips. I said at the beginning of this presentation that I don't really like people using equipment such as this anymore, because it gives the impression that electrochemistry is this big bulky subject, or electroanalytical chemistry is this big bulky subject. It's not. It's quite an elegant art. But if you do have, let's say you walk up to a potentiostat and it's got all these cables all over the place, the blue one's the reference, the red one's the working, and the black one's the counter. And we do use the same protocol, and we try to be consistent with that as well with our cables as well. So whenever you look at our screen printed electrodes, and I've noticed that a lot of manufacturers do the same, that we have a certain sequence of our reference electrode, our working electrode, and our counter electrode, and I know that other manufacturers also kind of keep to this the same color code and the same sequence of electrodes as well. Another comment is that people like to use glassy carbon electrodes a lot in R&D. They're interesting, but the problem for me is they put a barrier between an idea and a practical product. So this is a glassy carbon electrode. They're sort of $200 plus. You will actually find that electrodes from people like Zimmer and Peacock, they're really high quality anyway. So you can get publishable quality data out of a screen printed electrode, and you don't have to use the glassy carbon electrode. And my only problem with glassy carbon electrodes is almost philosophical, that it gives the impression that electrochemistry is a lab technique, when really, once you start using screen printed electrodes, you can start quickly imagining making products from these, products that people could actually buy. And so the screen printed electrodes from ZP, for example, especially our carbon electrodes, we can sell them at less than 50 cents each. So for the case of a glassy carbon electrode, you can get hundreds of these screen printed electrodes, which is also useful when teaching. And the quality of the data off a screen printed electrode these days can be just as high as a glassy carbon electrode. So we've done the sort of classic characterization of our electrode, shown that the peak height and the peak positions are fairly close to what the literature recommends. I say fairly close because sometimes we're sort of 10 millivolts off what the literature says is perfect. but I prefer the low cost and the ease of productization. And if I have to give up a little bit of perfection for that, then so be it. Let's say. Also, so I'm giving some practical tips and tricks. This happens a lot. I see this a lot with commercial screen printed electrodes. I don't see it with ours, but I do see it. People are doing cyclovoltammetry with these electrodes, and they actually see these really sharp peaks. If you see these really sharp peaks on a screen printed electrode, it's because you've got silver contamination on the working electrode. And it's very hard to get off. You have to basically complain to the vendor. So if you see these two sharp peaks in your voltammetry and you're not sure where they're from, and they're often symmetric around zero, then it's probably silver contamination and you have to complain to the vendor. You won't get it from us, but I have seen it quite a lot. So be careful of these two sharp peaks because they're silver contamination. They'll confuse you what you're looking at, and as I say, it's silver. At ZP, we do produce gold electrodes. We also produce a wash solution because gold tarnishes quite quickly. So if you're using gold screen printer electrodes and you're getting signals that look horribly and distorted, and you're expecting something to look beautiful, the gold is probably tarnished, and you need to activate that gold, and we clean that gold before we actually use it. We do have activation steps that we use internally. We talked a lot about cyclovoltammetry today. Cyclovoltammetry is useful because when you're doing you take an electrode, you add a material to the surface, then you add an antibody to the surface, then you put a blocking material on the surface. You can use cyclovoltammetry to actually show you how the surface is changing. And you would expect to change. You'd expect the cyclovoltammetry to become uglier and uglier and uglier, or more distorted, the more layers you put upon it. So if you see a distortion in your cyclovoltammetry at the very beginning, it tells me that your surface is some way unclean or blocked and it needs to be activated. I have received data from scientists in the past, whoopsie-daisy, where the cyclovoltammetry was extremely distorted. And the reason being is they were home making their electrodes. And they were making sort of carbon electrodes on paper. But the carbon was so resistive, they weren't getting the expected beautiful cyclovoltammetry. They were actually getting very distorted cyclovoltammetry. And it's because the resistance of the electrodes was just too high. And rather than getting nice peaks, it was stretched. And it was stretched because there was such a resistive element in there. It wasn't acting like an electrochemical circuit. It was acting just like a resistor. And when your sensor is acting just like a resistor, V is equal to IR, which kind of just says that you were just acting like a big resistor. So it gets very stretched in accordance with Ohm's law. Something that happens quite a lot in electrochemistry is you can expect nice signals and then you get this kind of high, get all this high frequency. I see this all the time. And it's just basically often just poor connections. There's no reason why electrochemistry should give you signals like this. When you get big jumps like this is often because somebody's moved the cable. And if you get sort of noise like this, it's because there's probably a poor connector in place. And so people send it to me and say, oh, look at this is terrible. It's just your connector's no good. So be careful for that. Poor connection can also lead to distortion. So if somebody was expecting some beautiful cyclovoltammetry, we thought their connector was bad. We sent them a connector. This is one off our web store, and suddenly they had beautiful results. And so this was what they expected. This was what they happened. And the only difference between the two is we were able to get back to perfection once we gave them a decent connector. I actually have other slides after this, which is really about the commercialization of things like academic research. But I want to sort of stop with that because I think this has been approximately 1 hour. So let me do a quick summary. So this webinar is about, in the end, we brought it down to electroanalytical chemistry, but this is a lecture series that we give at workshops. We also give it at Swansea University as part of our duties at Zimmer and Peacock to lecture to the undergraduates at the university or in the chemistry department. We've covered the techniques of voltammetry. We've covered the technique of potentiometry, amperometry, and impedance spectroscopy. And then we've just given you a few tricks and tips about trying to get quality in your data and avoiding things like bad connections or moving cables or dirty electrodes, or using things like glassy carbon electrodes, which I think kind of holds back the whole commercialization. So I hope that was interesting. If you've got any questions, reach out to us at Zimmer and Peacock. And if you're thinking about joining Swansea University, for example, I wish you excellent luck with it. Okay, thanks very much.